Hi everyone, and welcome to our module on autonomic receptors. In this video we're going to talk about the different receptors used by the autonomic nervous system, but before we get to that, let's talk about the neurotransmitters that are used by the autonomic system. As you know, if you've watched the video on the autonomic nervous system in general, the two major neurotransmitters are acetylcholine and norepinephrine. Both of these neurotransmitters are found in nerve endings stored inside membrane-enclosed structures called vesicles. This makes them ready to go anytime the nerve depolarizes and wants to transmit these neurotransmitters into the synapse. If the nerve depolarizes, this will lead to calcium influx, and it is that calcium influx that triggers the release of neurotransmitters into the synapse. Let's take a look at this picture on the screen here. We've got a neurotransmitter. Those are the green dots stored inside a vesicle. When the neuron depolarizes, voltage-gated calcium channels will open and allow calcium to come inside of the neuron. That triggers the synaptic vesicle to fuse with the membrane of the neuron. This is a process called exocytosis, and then the neurotransmitter is dumped into the synaptic cleft where it can go and bind to receptors on the postsynaptic neuron. For step one, you need to know a little bit about how the two neurotransmitters are synthesized. Let's start with acetylcholine. Acetylcholine is synthesized from choline and acetyl-CoA, and you may know if you've watched some of the biochemistry videos that acetyl-CoA is used in many places in cellular metabolism. So the enzyme choline acetyltransferase can take choline and acetyl-CoA and convert it into a molecule of acetylcholine. Now, once acetylcholine has been released into the synaptic cleft, it is exposed to another enzyme called acetylcholinesterase that breaks down acetylcholine. What this enzyme does is convert acetylcholine into choline and acetate. So acetylcholine is synthesized and stored in vesicles as we just discussed. It's released into the synapse, and then acetylcholinesterase breaks it down within the synapse. And here's my own little drawing of this whole process. Choline plus acetyl-CoA is combined into acetylcholine by the enzyme choline acetyltransferase. The acetylcholine is then packaged into vesicles. When the nerve depolarizes, calcium comes into the cell and causes acetylcholine to be released into the synapse, where it can bind with a muscarinic or nicotinic receptor. Also, while in the synapse, it can be broken down by the enzyme acetylcholinesterase into choline and acetate. And the one clinical tie-in of this whole process that you should definitely know is botulinum toxin, which causes botulism. I talk about this in the infectious disease videos, but this toxin can inhibit the release of acetylcholine from nerves that innervate muscle cells, and that's why it can cause paralysis. That's the dominant feature of botulism. Moving on, let's talk about norepinephrine. Norepinephrine synthesis begins with the amino acid tyrosine that is shown at the bottom of the screen. And in the biochemistry section, I have an entire video where I discuss tyrosine as an amino acid. For our purposes here, though, you should know that it is first converted to a structure called DOPA. DOPA stands for dihydroxyphenylalanine, and then DOPA is converted to dopamine, and dopamine can be converted into norepinephrine. And all of these changes from tyrosine to DOPA to dopamine to norepinephrine basically involve small modulation of the same molecule that begins with tyrosine. And here's my own little drawing of how this process works. It begins with tyrosine, which is converted to DOPA, and then dopamine. Dopamine is packaged into vesicles, it becomes norepinephrine, and when the cell depolarizes, norepinephrine is released into the synaptic cleft where it can bind to alpha or beta receptors. There's also a transporter which reuptakes norepinephrine from the synaptic cleft. That leads to it being repackaged so it can be used again. Because this process is the main way by which the sympathetic system exerts its effects on tissues and organs, there are a number of things that can modulate it. First of all, there are alpha-2 adrenergic receptors found on the presynaptic neuron. That's this neuron right here. These act as negative feedback regulators. So when there's lots of norepinephrine in the synaptic cleft, it binds to alpha-2 receptors, and that slows this process down so that norepinephrine release decreases. And this has clinical relevance because there are drugs that are alpha-2 agonists, and what they do is stop the release of norepinephrine. In the renal videos, I talk extensively about the renin-angiotensin-aldosterone system. That system produces angiotensin II, and it can modulate the sympathetic nervous system activity through A2 receptors. Angiotensin II receptors are found on presynaptic neurons, and they can increase the release of norepinephrine, and this ties the RAS system to the sympathetic nervous system. The parasympathetic system, when it becomes active, can shut down the sympathetic system, and the way this works is there are muscarinic M2 type cholinergic receptors found on the presynaptic neuron. When they are activated by the parasympathetic system, that decreases the release of norepinephrine and therefore dials back the sympathetic nervous system. If you have a time machine and you could go back in time to 1960, you would find doctors prescribing a drug called reserpine for hypertension. This drug inhibited the vesicular monoamine transporter VMAT, which packaged dopamine into vesicles, and it used to decrease sympathetic nervous system activity and lower the blood pressure. Other drugs that affect this system include amphetamines. Amphetamines are stimulants. They're sometimes prescribed to treat certain medical conditions. They can also be drugs of abuse. I talk about them in detail in the psychology section. But for our purposes here, they increase the amount of norepinephrine in synapses through a couple of mechanisms. First of all, they increase norepinephrine release. They also inhibit its reuptake. So through both of these mechanisms, they increase the amount of norepinephrine found in the synaptic cleft. Cocaine and tricyclic antidepressants can also inhibit the reuptake of norepinephrine. Tricyclic antidepressants are used to treat depression. Cocaine is a drug of abuse. Both of these can increase the amount of norepinephrine because they block its reuptake, and that's how they exert their effects. I'll go into a little bit more detail about amphetamines because these drugs have multiple effects on this system, and they like to test this on step 1. So as I said before, amphetamines are stimulants. They're also drugs of abuse, and they increase the dopamine and norepinephrine activity in the central nervous system. That's how they function. They do this through 3 mechanisms. First of all, they compete for the reuptake transporters, and therefore they inhibit the reuptake of norepinephrine and dopamine. They also can displace dopamine and norepinephrine from vesicles inside the neuron into the cytoplasm, and when that happens, the dopamine and norepinephrine in the cytoplasm get transported into the synapse. Let's go back to this picture here, and let's imagine that we're looking at a neuron in the central nervous system. Amphetamines compete for this reuptake pump here, and therefore less norepinephrine or dopamine is taken up, and therefore more of it remains in the synapse, and what you get in the brain is generally more norepinephrine or dopamine activity. In addition, once amphetamines do get taken up inside the neuron, they get put inside of vesicles instead of norepinephrine or dopamine. This leaves more norepinephrine and dopamine floating around in the cytoplasm of the neuron. When the level of these substances gets high, they then get dumped out into the synapse. So the net result is that you block the reuptake and you increase the release of norepinephrine and dopamine into synapses in the brain, and that's how amphetamines exert their effects. Now let's move on to the main focus for this video, and that is a discussion of autonomic receptors and how they work. As you know, if you watched the video on the autonomic nervous system, there are 3 types of receptors found in the autonomic system. There are nicotinic receptors, which bind acetylcholine, and therefore they are also called cholinergic nicotinic receptors. There are muscarinic receptors, which also bind acetylcholine. These are called cholinergic muscarinic receptors, and there are several subtypes M1 through M5. And then finally there are adrenergic receptors. These bind to norepinephrine, and there are alpha-1 and alpha-2 receptors, as well as beta-1, beta-2, and beta-3. Here is our map of the autonomic nervous system, which I go over in detail in the general video on the autonomic system. For our purposes here, we're going to talk about the receptors, and I'm going to begin with the nicotinic receptors. So nicotinic receptors are always found in ganglia of the autonomic nervous system. They're found in the ganglia of the parasympathetic system, as shown here, and they're found in the ganglia of the sympathetic system, shown here. They're also found at the adrenal medulla, and they are also found at skeletal muscle. The way skeletal muscle is innervated is a single motor neuron goes to a skeletal muscle cell, releases acetylcholine onto a nicotinic receptor. Nicotinic receptors are different from all the other receptors we're going to talk about in this video, and that's because nicotinic receptors only do one thing. They cause depolarization of another structure, either a muscle cell or a nerve. Therefore, nicotinic receptors are always ion channels that allow positively charged ions to enter a structure and depolarize that structure. Nicotinic receptors are described as ligand-gated ion channels, and this just means they are ion channels that can open up like a gate when they bind to a ligand, and the ligand in this case is the neurotransmitter acetylcholine. So let's look at this drawing on the bottom of the screen. We've got a molecule of acetylcholine coming in to bind to the nicotinic receptor, which is the ligand-gated ion channel. Out here floating around we have ions like sodium and potassium, and even calcium and magnesium. When acetylcholine binds, the channel opens up like a gate and all these ions pass through, and that's going to depolarize either a neuron or a muscle cell. That's how these work. So going back to this map, you find nicotinic receptors anywhere the nervous system needs to cause depolarization. So in the parasympathetic system, we need to cause depolarization of the postganglionic neuron, therefore you find a nicotinic receptor. You also find them here in the sympathetic system where we want to cause depolarization. You also find them on skeletal muscle cells where we want to cause the skeletal muscle to depolarize. Anytime you need to depolarize something, you're going to find a nicotinic receptor. That's because it is a ligand-gated ion channel. All the other receptors in the autonomic nervous system, like the muscarinic receptors and the adrenergic receptors, have a very different structure, and that's because these receptors are not built to depolarize anything. They're built to generate an effect, for example, to contract smooth muscle or cause the salivary glands to produce more saliva. So they have a different structure called a G protein coupled receptor, or a GPCR. These receptors get their name because they are protein receptors attached to guanine nucleotides. Guanine nucleotides are molecules like GDP or GTP. They are similar to ATP. They are nucleotides inside of cells. And once the G protein coupled receptor binds its ligand, in other words, binds a neurotransmitter, then it activates a second messenger. A second messenger is a molecule that can go on and create the desired effect, for example, smooth muscle contraction or secretion of saliva. And here I've zoomed in on the picture from the last slide. So a neurotransmitter is binding to a G protein receptor. That G protein receptor contains GDP, which is a guanine nucleotide, and that's why it's called a G protein receptor. Once the neurotransmitter binds, the G protein modifies the level of some second messenger molecule, which then goes on to carry out the intended action of the autonomic nervous system. So what are the second messengers used by the autonomic nervous system? There are only two of them. The first one is cyclic AMP. The second one is inositol triphosphate, or IP3. I've shown their structures on the screen here. Cyclic AMP is a molecule of adenosine monophosphate, but the phosphate bond makes a circle here. That's why it's called cyclic. Inositol triphosphate also has a ring structure, and it's got 1, 2, 3 phosphate groups. That's why it's called inositol triphosphate. So anytime one of these G protein coupled receptors is activated, it is going to increase or decrease the levels of either cyclic AMP or IP3 inside a cell, and that will create the desired effect of the autonomic nervous system. Cyclic AMP is synthesized by an enzyme called adenylyl cyclase. It is synthesized from molecules of adenosine triphosphate, or ATP, which you probably know is a major energy source inside of cells. So what the G protein coupled receptors do is they modulate the activity of adenylyl cyclase, either increase or decrease it. That leads to generation of more or less cyclic AMP, and then cyclic AMP is the second messenger that carries out the desired effects of the autonomic nervous system. Inositol triphosphate is synthesized by an enzyme called phospholipase C. What it does is it takes large molecules, large fatty acid type molecules called PIP2, or phosphatidyl inositol biphosphate, and converts them into IP3. So once again, what the G protein will do is activate phospholipase C. That will make more IP3 inside of a cell, and IP3 will go on to carry out the desired effect of the autonomic nervous system. So everything I've told you up to now is fairly straightforward to remember and understand. What gets very confusing for medical students is that there are a number of subtypes of G protein coupled receptors, and they all have different effects on second messengers. The three subtypes are called GS, GI, and GQ. They all have either different second messengers or different effects on the second messengers. The GS subtype are found in beta 1 and beta 2 receptors. Anytime you hear of a beta receptor, it's going to be a GS subtype. The GI subtype is found in the M2 receptors, and the GQ subtype is found in alpha 1 and M3 receptors. In the next few slides, I'll go over how these different subtypes work and try to give you a framework to remember why different types are found associated with different receptors. The GS subtype of G protein receptor gets its name because it stimulates adenylyl cyclase, and as we talked about before, when this enzyme is activated, that will raise the intracellular level of cyclic AMP. This is depicted in this cartoon on the screen. We've got a neurotransmitter binding a G protein receptor. The G protein receptor has a number of subunits that aren't important for you to know, but the alpha subunit is bound to GDP. That's what makes this a G protein receptor. Once the neurotransmitter binds, the enzyme adenylyl cyclase is activated, and that converts ATP into cyclic AMP. Cyclic AMP has different effects depending on whether it's found within cardiac myocytes or vascular smooth muscle. In cardiac myocytes, high levels of cyclic AMP are stimulatory. For that reason, beta-1 receptors found in the heart are the GS subtype. Anytime norepinephrine binds to a beta-1 receptor, it will raise the level of cyclic AMP, and that will activate cardiac myocytes, and that will raise contractility. In smooth muscle, cyclic AMP causes relaxation, therefore beta-2 receptors, which as you may know are the receptors that cause relaxation of smooth muscle, for example in the bronchioles of the lungs, these all have the GS subtype. So an easy way to remember this is that all beta receptors have the GS subtype of G protein receptor, and activation leads to higher levels of cyclic AMP. The GI subtype of G protein receptor inhibits adenylyl cyclase. That's why it's called the GI subtype. This is going to decrease the intracellular level of cyclic AMP. In cardiac myocytes, that means that when this type of receptor is activated, that will decrease contractility, and therefore you find this type of receptor in M2 receptors. M2 receptors are part of the parasympathetic system. Their goal is to decrease contractility of the heart, so they are associated with the GI subtype of G protein receptor, and they lower cyclic AMP levels. You can also find the GI subtype of G protein receptor in those alpha-2 negative feedback receptors that I talked about earlier in this video. So here's a little cartoon of a cardiac muscle cell on the left and a vascular smooth muscle cell on the right, and I've depicted how the different G proteins work on these two types of cells. Let's start with the cardiac muscle cell on the left. Here is the enzyme adenylyl cyclase. Beta-1 receptors will activate this enzyme through GS proteins. M2 receptors will inhibit this enzyme through GI proteins. That enzyme increases the level of cyclic AMP, which can activate the enzyme phosphokinase A. Phosphokinase A causes more calcium to come into the cell, and calcium stimulates the sarcoplasmic reticulum to release more calcium, and this causes more contraction. So this is how the two different G protein receptors affect the level of cyclic AMP, and how cyclic AMP leads to more muscle contraction. In smooth muscle cell, we find generally only GS proteins. They are linked up with beta-2 receptors. This activates adenylyl cyclase and raises the level of cyclic AMP. What cyclic AMP does in vascular smooth muscle is inhibit the enzyme myosin light chain kinase, and when it is inhibited, there is less contraction because myosin light chain kinase's normal role is to interact with calmodulin and calcium to cause contraction. If you find all this confusing, just remember what I showed you on the last two slides about how the two different G proteins affect cyclic AMP and what cyclic AMP does to cardiac muscle and vascular smooth muscle. The GQ subtype is the only subtype that activates phospholipase C, and therefore it raises intracellular levels of IP3. This is shown in the cartoon on the screen here. We've got a neurotransmitter binding to a GQ subtype G protein coupled receptor. That will activate phospholipase C, which will convert PIP2 into IP3. In addition to IP3, it also makes another molecule called diacylglycerate, or DAG. The level of IP3 will then go up within the cell, and that's the second messenger which generates the effects of the G protein coupled receptor. In terms of the autonomic nervous system, GQ subtype G protein receptors are found only on vascular smooth muscle where they cause contraction. So they are going to be found associated with any receptor that leads to contraction of vascular smooth muscle. In the sympathetic system, this is the alpha-1 receptors. These are the classic vasoconstrictor receptors of the sympathetic nervous system. They are going to be linked to the GQ subtype of G protein receptors so that they can contract vascular smooth muscle. In the parasympathetic system, the GQ subtype is linked to M3. M3 receptors are the receptors in the parasympathetic system that in some cases cause vasoconstriction. They can cause bronchoconstriction in the lungs, they can cause contraction of the detrusor muscle in the bladder, and they can also cause contraction of the ciliary muscle in the eye. And here's my own drawing giving you a little bit more detail of how the GQ systems work. So when these types of G protein receptors are activated, they activate phospholipase C, which increases the level of IP3. IP3 causes the sarcoplasmic reticulum to release more calcium, and that causes contraction of vascular smooth muscle. So I've indicated at the bottom a key point to remember: GQ is found only in vascular smooth muscle, and it always leads to contraction. So G protein receptors are found in the autonomic nervous system when that system wants to carry out effects like vasoconstriction or bronchodilation, but they're also found in neurons throughout the brain and other parts of the body where they aren't necessarily carrying out some mechanical effect like constriction or relaxation. So shown on this slide are all different types of receptors in the body that are linked to G proteins. So the alpha and beta receptors and the muscarinic receptors are linked to G proteins, but so are the dopamine receptors, histamine receptors, and vasopressin receptors. All of these are linked to some of the different subtypes of G protein receptors. And if you want to remember all of these, there is a mnemonic that most medical students learn. The GQ subtype of receptor can be remembered by the mnemonic HAV1M&M. That's because the H1 histamine receptors, alpha-1 receptors, V1 receptors, M1 receptors, and M3 receptors all are linked to GQ proteins. The GI subtype can be remembered by the mnemonic MAD2. That's because M2, alpha-2, and D2 are all linked to GI proteins, and then all the others are found associated with the GS subtype. Use this if it helps you. If it doesn't, memorize it any way that you wish. And that concludes our module on autonomic receptors.